Hi, everyone. Welcome. Today, we're having Gene Therapy and Neuroscience 101 as part of the Harvard Brain Science Initiative's 101 series. Today, we're really excited to have with us Doctors Connie Cepko and David Liu. And I'm just going to do some brief intros of both of them together so that we can seamlessly transition from one to the other talk. So Dr. Cepko is a geneticist and neurobiologist. She is the Bullard Professor of Genetics and Neuroscience at Harvard Medical School and an investigator of the Harvard Hughes Medical Institute. Currently, her research is focused on the development of the central nervous system and CNS diseases. She's particularly interested in the retina. Her lab is developing gene therapy methods to prolong vision in genetic forms of blindness. They've used genes that fight oxidative stress, inflammation, metabolic problems, to prolong vision in mouse models of inherited retinal degeneration. Her lab also specializes in developing a variety of tools for neuroscience research. And I'm happy to note she's on the steering committee for the group sponsoring this, the Harvard Brain Science Initiative. Dr. David Liu is a chemist and chemical biologist. He is the Richard Merkin Professor and Director of the Merkin Institute for Transformative Technologies in Healthcare, Vice Chair of the faculty at the Broad Institute, and the Thomas Dudley Cabot Professor of the Natural Sciences at Harvard, and also an HHMI investigator. His research integrates chemistry and evolution in order to illuminate biology and enable next generation therapeutics. Three major interest areas of his lab are, as you'll hear about today, gene editing, protein evolution and delivery, and DNA templated synthesis, which is the discovery of bioactive synthetic small molecules and synthetic polymers using DNA templated organic synthesis and DNA encoded libraries. So we're very happy to have both of you here today. And what we'll do is Dr. Cepko will speak and then Dr. Liu will speak, and we'll have about 30 minutes for Q&A. So thank you again, Dr. Cepko, for getting us started.
Well, thanks very much, Perry, for organizing this and then for inviting me to participate. It's always fun for me to talk about gene therapy vectors. I only have 30 minutes, so I'll be not able to cover all the things I like to, but I hope in the questions we can address some of the missing points. So I'll give you a little bit of overview of some sort of high-level principles that one considers for gene therapy, and then a bit about some of the viral vectors and some current applications. So first, just a definition, because I think this term is somewhat ambiguous and it's a field that's advancing rapidly. So we have all different sort of concepts about what it can be, but I just make it fairly simple and say that it's changes in the genetic material of a host to achieve a therapeutic effect. Now, the genetic material can have various forms. We can have double-stranded DNA, single-stranded DNA, messenger RNA, bits of RNA, and all sorts of combinations. The goal then, though, is to change something in the host using those genetic materials to combat some type of disease process. So there's a couple approaches that we can consider. One, and the most obvious, and the first one that was deployed, is gene augmentation. This one's kind of obvious. If you have a loss of function allele in a gene that leads to a disease phenotype, and you add back a wild type or a good copy of that bad gene, you can then hopefully address some of the limitations of that loss of function and thereby get a therapy. One might also have other alleles which cause negative effects, so dominant negative or other types of deleterious effects. And in that case, rather than augmenting, one wants to knock down the responsible gene. And that can be done, as David will tell you, through some gene editing approaches or even shRNAs, and those are being used in the clinic now. David's going to cover gene editing, but one can consider all sorts of ways that one can address then problematic alleles in the genome directly with gene editing. And one can also add a novel function or a gain of function. For example, for immunotherapy, one can deliver a chimeric receptor, which allows T cells to more effectively kill tumor cells. So all of these could be considered forms of gene therapy. So if you want to launch a gene therapy, or you're thinking about it, you're just generally interested, there are a number of considerations. Some of them are kind of obvious and some are not. One that would have seemed to be obvious and simple is just understanding the disease biology. It turns out that not every disease can be approached with this type of therapy. You really have to understand something about the progression of the disease, the target cells for the disease, the type of problems created by the disease gene. And so more knowledge of the disease biology is extremely helpful in terms of targeting the right time, the right cells, right number of cells, and that sort of thing. Availability of a therapeutic gene is obvious if one is looking at a loss of function and gene augmentation, but we have a lot of diseases, including those that are pretty common, where we don't have a simple Mendelian inheritance pattern or something that's obvious in terms of a therapeutic gene. And in those cases, what we're dealing with is symptoms. And it's very helpful if we have some type of animal model to run through some candidates that might be therapeutic. Something that I think is super important, but it's not always up at the top of people's list, is this business of the target cells being available for the transduction. So infection with a viral vector or transduction with a nanoparticle. The target cells have to be available, and they have to be infectable in sufficient numbers for a therapeutic effect. There are a lot of target cells which are widely disseminated in the body, and they are present in very large numbers. For example, skeletal muscle, brain neurons. Those are not readily available for a direct inoculation or direct infection. And so one has to consider carefully what one needs to deliver in terms of the number of cells that have to be transduced for a therapeutic effect in such cases. So target cell availability is huge. Then, of course, one has to have a vector, and the vector has to be able to accommodate the gene. And as you'll hear from David, that's not so straightforward for some of the common vectors in use when one is considering large genes, as in the gene editing machinery. But even many disease genes don't fit in some of the more popular vectors. Now, this next one seems maybe, again, kind of trivial, but if you're in the vector business, it's not. And one has to be able to make enough vector. Now, it turns out that that's not trivial for many types of vectors. Viral vectors, as I'll go over, lend themselves to this because they actually replicate. But especially if one is targeting a large and disseminated number of target cells, again, let's just use the example of skeletal muscle, you have to be able to grow a really large amount and have it be very concentrated. That puts a lot of pressure on the manufacturing process, and also in terms of making it very pure, because if you're injecting a lot of stuff, then you don't want any kind of toxic byproducts in there. And lastly, one has to consider the immune response. So viral vectors and even nanoparticles can stimulate the innate immune system, which can kick off a response, which, if it gets out of control, can create what's called a cytokine storm. In fact, sadly, that did end up killing one of the early gene therapy recipients in the mid-90s. So we can have immune responses just to the viral capsid or something inside the capsid. Also, if we're targeting a null mutation, so the body's never seen a particular protein and we're doing a gene augmentation to add it back, there may be a problem with that being recognized as foreign. And if a person has had prior infections with a virus that's very much like one of the vectors, there can also be immune responses, for example, neutralizing antibodies or T cells, which can kill infected cells. So prior infections for some of the common vectors is something that's also under consideration. So there's a lot that needs to be considered. And I would say for me, the most important to start off with is target cells. Are they available? My computer's freezing. Oh, there we go. Okay, so what kind of vectors are available? So viruses and nanoparticles. And I think of nanoparticles kind of as sort of virus wannabes. They take a lot of the strategy that's evolved for viruses to deliver nucleic acids. And there's some fundamental differences, however, and I'll mention those. But this is a very rapidly evolving area where people have all different kinds of formulations. It's really interesting how creative people are being in terms of making nanoparticles that have different moieties on the surface that might target or might give the virus access to certain cell types, as well as formulations for the nucleic acid so that they're more long-lived. But generally, nanoparticles are encapsulating a nucleic acid like a messenger RNA, whereas viruses have more flexibility in terms of different kinds of nucleic acids that they can deliver. So just to summarize for nanoparticles, because they often are carrying something like a messenger RNA or even a DNA molecule, they have not evolved the type of features that viruses have in terms of getting their genome stably expressed for a long period of time. So they're typically used when one wants transient expression, again, as in gene editing, Whereas stable expression, people do not use nanoparticles, they use viruses. The cargo capacity for nanoparticles and the type of cargo, though, is quite flexible. They don't have a hard capsid that's evolved to house a certain size of genome, and viruses have done so. So the cargo capacity, the type of cargo, and again, the particle itself, there's a lot more flexibility. So that flexibility is being exploited by engineering, again, different kinds of surface molecules and even things inside the particle. Some of this is for targeting, some of this is for longevity of expression, some of this is for accessing different aspects, let's say if it goes into the bloodstream, getting out of the bloodstream into some target cells. So a lot of engineering, a lot of activity in this area, some of it extremely creative. But the viral vectors are the ones that have been used most so far for gene therapy. And I just want to go over why these have been sort of the leading vectors in this space. But I'm going to take a quick break. My puppy is crying in my office, and I have to put him outside my office for a minute. Be right back. Okay. All right, sorry about that. He's very cute, but he's needy at this stage in his life. He's only 4 months old. Okay, viral vectors, my favorites. So why do we use them? Well, they have evolved to transduce genomes, their own genomes, and to replicate them. So they're really the professionals for gene transfer. What this does, it allows us to, for example, manufacture more easily because they replicate. So if one can get the replication started, and there's all kinds of tricks to do that, you can grow some of them to very high titer or concentration and very high amounts. Some of them are also very efficient at infection. So if you put a preparation on a group of cells, a lot of them can be transduced because, again, they've evolved to do that. Now, among all the viruses that we know about, there's a range of features, and I'll tell you a little bit about them, but let's just say different kinds of genomes, different life cycles, some for integrating, some for not integrating. So these features have been exploited, and there's lots more out there that we haven't exploited. And I would say that we need to really take a hard look at some of the viruses that we're not using, because the current crop of viruses don't really fulfill all our needs. They can be engineered. The genomes are easily engineered, typically using molecular biology. But one can also engineer the capsids or the shells, the lipid envelopes around some of the viruses to get, again, features mostly for targeting specific cell types. Some viruses are stably associated with the host cell for the life of the cell and all of the daughter cells. Those are stably associated genomes that I'll tell you about. And just one thing I want to put in a plug, because people often think that they can't make vectors in their lab. But in fact, you can buy vectors. You can pay companies to design them and produce them and give them to you. This costs a lot. It also takes a lot of time. If you have a tissue culture hood and you know how to do molecular biology, it's really not hard to make AAV and lentiviruses, the current ones that are in use for most gene therapy trials. So I encourage anybody who wants to try this out to not be too shy about trying it. There's lots of great protocols online, and there's a lot of help available. So you can do this. For the nanoparticles, though, I see when I look around that to make the really advanced ones takes more expertise, and it's better to just collaborate. Okay, so this is a little list of some of the viruses that have been used. It's not complete, but these are the more popular ones. So adenovirus was one of the earliest ones. We understand it pretty well because, again, it replicates really well. It's one of the ones that was first studied in terms of its molecular biology. It's a virus that has no plasma membrane, so it's just a protein shell. Its claim to fame as a vector is that it has a very large double-stranded DNA genome. It's 36 kb. You can delete all the genes and then use that space for putting in regulatory elements in multiple genes. It's also good at attracting the attention of the immune system, the innate immune system, which makes it inflammatory. That's useful for vaccines, and it is being used for a SARS-CoV-2 vaccine, but it's not good for gene therapy. You can get a lot of inflammation from delivering adeno, and I mentioned that there was a cytokine storm that killed an early patient, and that was from a high dose of adenovirus. So we need to learn how to tame this thing in terms of damping down the innate immune response. But I do think it has good possibilities because of this large cargo size, and it grows really well, and it's stable. Adeno-associated virus is one that was found actually in association with an adenovirus long ago. And it's a non-pathogenic virus, and it's got a small genome, only about 4.5 kb. It's pretty simple to grow and easy to grow. It's also stable. It also doesn't have a lipid shell, and that tends to make it more stable. And I'll come back to other features that make it pretty popular. And then there are retroviruses. So these are really useful if one has a need for stable genome association of the vector with the host. And I'll talk more about that, but these are two types that we use, gamma retroviruses and lentiviruses. Lentiviruses are actually retroviruses, and the reason they're useful is they integrate their DNA into the host chromosomes. They have about an 8 to 9 kb cargo size. So I'm just going to tell you a little bit about the life cycles of these viruses so we can see why they're being chosen in a couple applications. So this is a little diagram of a lentivirus. So the genome in retroviruses is RNA. So there's 2 molecules of RNA as a genome. It's surrounded by a nucleocapsid that protects the nucleic acid. Around that is another capsid. And then this one is what's called an enveloped virus. It has a plasma or lipid envelope around it, a plasma membrane it actually stole from the host cell that produced it. And then a feature that makes it particularly versatile is there's a glycoprotein embedded in that envelope. And that glycoprotein determines the host cell receptor binding and entry. And what's nice about a lot of these enveloped viruses is you can swap in and out these different glycoproteins. And the one that's typically used for vectors, all the retrovirus vectors, is from the vesicular stomatitis virus. It's called VSV-G. And the reason we put G on the surface rather than the endogenous retroviral glycoprotein is that that G gets this particle into virtually every cell type. So entry is no longer restricted in any way. It also just happens that it's stably associated with the envelope, in some cases more stably than the normal glycoprotein. So these lentiviral particles then have a very wide host range. And again, they have about an 8 to 9 kb cargo capacity. So the life cycle for retroviruses is that they will get into the host cell by binding to a receptor, and then the lipid envelope or plasma membrane of the virus can fuse with the host cell plasma membrane, or in some cases, they can enter and fuse with an endosomal membrane. That liberates the capsid to the cytoplasm, and it kicks off reverse transcription. So retroviruses are famous because they reverse transcribe, and in fact, the reverse transcriptase that you use in your lab came from a retrovirus. And that makes a double-stranded DNA copy, a very precise copy of the viral genome. And the really important thing is that that viral genome can then enter the host nucleus and integrate into any one of a very large number of sites, mostly into active chromatin. And that integration is efficient and precise. So the ends are exactly as they should be for the virus to be able to express all its genes. So it's not scrambled. You get one copy integrated per infectious viral particle. And it's stable. It's there forever for the life of this cell and all its progeny. Now, normally what happens after that is viral RNA is made. So transcription starts after integration. Viral RNA is made, proteins are made, particles are made, and more virus is produced. But in all the viral vectors, lentiviruses included, that process, that process is interrupted. There is no replication because all the genes for replication have been deleted. One point I want to make about lentis versus gamma retroviruses is, again, all the retroviruses integrate, but the gamma retroviruses can only gain entry to the nucleus if the nuclear envelope breaks down during the M phase of cell cycle. But lentis have evolved a way to get themselves in through a pore. So you can get lentiviral integration into postmitotic cells. And that's the virus you would use if you wanted to have integration into any postmitotic cell, neurons, postmitotic muscle cells, et cetera, if you want integration. Okay, so that was a short rendition of retroviruses. So for AAV, this is a beautiful structure of the AAV capsid. AAV is a non-envelope virus. You're looking at protein. There's no envelope. So you can't put the G protein from VSV on an AAV to give it the really broad host range that lentiviruses have. So people are doing a lot of engineering of these capsid proteins, in fact, to create specific entry points into specific cells and perhaps other aspects that create more specificity for things like genome encoding or other aspects of the viral life cycle. So the way this one works, this is, I should say, it's a single-stranded DNA genome, again, a small one. So it can enter through a receptor. There's one receptor that's been defined that's used for almost all of these AAVs, but there are co-receptors also that can alter the specificity. They come in, get in through a clathrin-coated pit, which then ends up in an endosome. So the particle goes through, ends up in an endosome. It can then escape the endosome and get into the nucleus. And some of these steps are still a bit unclear precisely how they work. But once it gets in, that single-stranded genome then becomes double-stranded. And after that, it can transcribe. Now, as part of its normal life cycle, it does not integrate, but it can integrate. And in human cells, it integrates into one particular locus, at least in tissue culture. It doesn't seem to do that when one infects in vivo. Instead, if there is integration, it's not a very tidy process like it is with retroviruses. The viral genome can be interrupted. There can be duplications, inversions, all kinds of sort of messy genomic arrangements. And that's an aberrant process in terms of AAV. It normally stays episomal. That is, it does not integrate. It does not replicate when the host genome replicates. It does not in any way replicate itself so that it will get diluted out if a cell that's infected divides. That is, the daughter cells can't get genomes that have been replicated. So keep that in mind if you ever want to target a mitotic cell and have the genome or the products expressed by a lot of daughter cells. So the genome is really small and simple, and that's lent itself to some of the engineering that people do. So it normally has some of its own genes for replication. Those are taken out. One can put in a promoter of one's choice and a transgene. These promoters tend to be small because, again, the genome is quite small. One can make it even smaller, but a little more efficient if one creates a version called a self-complementary. This one, it goes in double-stranded, is only though about 2.5 kb. So most people are using these single-stranded versions, but you do have the option of using something that expresses a little more quickly, and that's called a self-complementary. So most of the AAV vectors that are in use right now have some type of specific promoter and a transgene, all restricted by size. So why is it so popular? So I should say it's not pathogenic. There's lots of them in nature to work with. So that I already mentioned that, but I'll say that again. And it can infect, transfect both dividing and non-dividing cells. And by some miracle of nature, when it gets into non-dividing cells, it just stays there for a very long time, maybe forever. There's now gene therapy in dogs and in people that go for 10 years. So after one inoculation, there's still gene expression from the AAV genome, which again is not integrated, it's just hanging out, and it's not being destroyed or silenced. And so that's just by good fortune. It can also infect dividing cells, but again, it will be diluted out with cell division. I mentioned this business about the host genome. This one happens to be pretty easy to produce at high viral titer, and it's not contaminated by helper viruses, which the early generations of viral vectors had an issue with helper viruses, that is, viruses that could replicate contaminating the virus stocks. Oops, this one does not have that problem. In general, but not completely true, it's not as immunogenic as some other viruses. I mentioned adenovirus. Adenovirus, again, is very inflammatory. AAV is much less so, but if you put in high dose, you can get inflammatory responses. And people have been using it for in vivo gene delivery. So now let's talk about how you use these viral vectors. How do you actually get delivery? I can divide that up into two strategies. One is to take out the target cells, so culture them ex vivo, infect them, test whether the infection worked, and then put them back in. So that would be an ex vivo strategy. One can also directly inject the person with the virus. Now, one has various reasons for choosing one or the other. A lot of cells, you just don't have the option of taking them out and culturing them. Let's say you had brain cells as your target. It's really hard to get them out, culture them, infect them, and put them back in and have them integrate, synaptically connect, et cetera. There's possibilities using organoid-derived neurons, but again, I will say that I think getting engrafted neurons to do their job is going to be tough. But there are other cells which you can culture quite readily and infect. I'll give you one example. And then it's possible to directly inject. And again, the crucial thing about that is, will the target cells get hit? Where do you inject? How much do you inject? And how does it get to those target cells? So for ex vivo infection and engraftment, the first gene therapy that was done, and one that's still ongoing, is to target the hematopoietic stem cells. I'm sure most of you know that the hematopoietic system has a lot of different cell types. They're affected by a lot of different disease genes, but they all come from what's called a long-term repopulating stem cell. And one cell can repopulate with all of these types of daughters. So it's an amazing process, actually, and it goes on during our whole lives. And you can culture these, and they will make various intermediates along the way. If you target some of the end stage cells, they are more short-lived. And so your gene therapy will basically be extinguished over time. And so people are going for these long-term cells because if one can get them transduced, then one has perhaps a lifelong replenishment of each of these types of cells, including then a therapeutic effect in the target, the end stage target cell. What that means, though, is this long-term cell is very mitotic, makes lots of divisions to create all those cells. So you have to use an integrating virus, and that means you have to use a retrovirus. So that's exactly what's being done right now. People take out bone marrow stem cells, they expose them to a retrovirus, and then they put them back in. The initial trials had some problems with viruses integrating and creating leukemias. That hasn't happened in a long time. People have redesigned the vectors. So hopefully this can continue to go on in a safe way. And there's a lot of different diseases being targeted. This is just some very simple, actually now out of date, but these are names of some diseases you may recognize in the target cells. Beta thalassemias, all kinds of sickle cell disease, various hemoglobinopathies are being targeted, as well as a whole bunch of other ones. And so the strategy has been established. It's actually rough on patients to go through these things, and it's extremely expensive. It's really not a scalable type of therapy, but it's actually one that can be effective. And the people who, for example, have been treated for sickle cell are very grateful. So for some people, lucky few, it works extremely well. Not sure it's going to be scalable. Okay, what about direct injection of virus? So one that's been FDA approved is called Luxturna. It's for a disease called Leber's congenital amaurosis 2. These are people that are born blind or almost blind due to a deficiency in an enzyme called RPE65. That enzyme produces the chromophore that allows our photoreceptors to capture light. So if you can't make that chromophore, you really can't transduce a light signal. This is being delivered by AAV, and it's a fairly simple procedure where the AAV is inoculated directly into the eye. And obviously the eye is very accessible for injections. And this space, although it takes some skill, is not super hard to hit. So very simple kind of operation, delivery is straightforward. And there's some features that have made this particularly amenable to this type of therapy. So one of the key things comes back to this target cell idea. So the virus is delivered right on top of the RPE cells, the cells that express the RPE65 gene. I can't emphasize this enough. There's nowhere for the inoculum to go. It does not go outside the eye. It only goes right onto the target cells. So this makes for less tighter requirements. You don't have to put in so much virus. In fact, let's say compared to some of the amounts that are being delivered systemically to hit muscle, there's 10,000-fold less virus being delivered for this therapy than for some of the muscular dystrophies. So that puts a lot less pressure on manufacturing, cost, and things like inflammation. So local delivery, efficient access, or efficient infection because the cells are very accessible from that inoculation site. The cells happen to be rather large. These RPE cells are big. They take up virus really well. We've done some quantification of how many viral genomes are in each RPE cell from what we do in mice, and it's at least 30 genomes per cell, perhaps again because they're really large, a lot of surface area. Now, a key thing is that the RPE65 I mentioned is making a small molecule chromophore. It gets secreted from the RPE cells, and one RPE cell actually can supply chromophore for about 100 photoreceptor cells. This again means that you reduce the pressure on getting a high frequency of infection, even at the inoculation site. If you only get a scattered infection, you can still have enough chromophore for a lot of adjacent photoreceptors to be able to detect light. So this secreted aspect and the fact that, again, this thing can diffuse and hit a lot of cells is probably one of the reasons why this is successful. There's also so-called immune privilege in the CNS, including within the eye. It's not clear to me that this is going to be true in diseased eyes, but generally speaking, there's not as much immune surveillance of the eye. It's sort of a closed system. It has what's called a blood-retinal barrier, and it doesn't have the same kind of T cell, B cell, et cetera, surveillance that goes on in the periphery. And I'll just mention that so far the safety profile has been extremely good, again, leading to its approval by the FDA. I'll mention one more thing, that there's a couple versions that have been made to deliver and express this. One specific promoter for the RPE65 gene, the endogenous promoter, and one that's actually generic, the CAG promoter, which many of you know about. And the one that's actually approved is the CAG promoter. So sometimes you can get away with using a non-specific promoter, and in other cases you want a specific promoter. And that goes back to what I said at the beginning, understanding the disease process and the requirements for the therapeutic effect. Another set of diseases that are being targeted, and this is again by in vivo delivery using AAV, are for hemophilia A and B due to loss of function in factor 8 and 9. So this affects the clotting cascade. One of the earliest gene therapies was for factor 9 using AAV. It initially ran into trouble because there were memory T cells in people that recognized viral epitopes on the surface of infected cells, and it killed them. The virus is delivered now intravenously, and when you put something into the circulation, it goes to the liver. It turns out the liver is the normal site of synthesis for factor 9. So just injecting systemically gets the target cells well exposed, and these AAVs are very efficient at infecting liver cells. So again, target cells are accessible, they're efficiently infected, and this is again the reason why this is inching towards FDA approval. It probably will be approved very soon. The trials have looked really good. So why is it successful again? Well, delivery and access, huge. This is absolutely crucial to success. People have been using different capsids. Many of them will get in. People are, in this case, using specific promoters. Now here we come back to the secreted product. So the factors get secreted by the liver cells. It turns out that you can have even just 2% or so of the normal levels of these factors in the blood, and people are still able to get off transfusions, which are the normal or the previous standard of care. So even if you don't have a high percentage of liver cells infected, you can still have a good therapy. So again, a secreted product and good access to target cells is probably what's going to make this the next FDA-approved therapy. So I'm running out of time. I'll just pick a few of these things to tell you. This is really the end of my talk. And if you're interested in using AAV, I've mentioned some of these, and I'll just go over them real quickly. Capsid choice is important. There's a whole industry of people in academics and companies engineering the capsid for targeting specific cell types. You have your choice of single or double-stranded genome, and that's going to be dictated by the size you need. People are generally using restricted promoters. They're going to be small. If the cell is mitotic, I would say don't use AAV. It will get diluted out unless you want transient expression, and then it's a good thing. For neuroscientists, it's kind of interesting. If you put in high doses of AAV into the nervous system, they can be taken up at axonal terminals and go retrograde. They can come from cell bodies and go out antrograde. This is pretty inefficient, but it's kind of interesting. Normally what's being delivered is to the cell bodies. If you're actually in this business, and I speak more to the basic scientists here, things like the rate of injection tissue density are important. Most of the infection happens along the needle track if you're going into a packed tissue. If you go into a lumen, you get more distributed infection, but the virus gets diluted. And some of these other things I already mentioned. And just for fun, I just show you there's tons of AAV capsids in nature, hundreds of them. So even without engineering, just by selecting the right one, you can get infection with different efficiencies in different tissues. So I think I'm going to stop there, and hopefully David can pick up and tell you how gene editing is now being used for gene therapy.
All right, great. That was really beautiful, Connie.
Thanks,
really,
Dave.
really clearly presented, and is very complementary to what I hope to share with this group, which is on gene editing. If I can get my slides shared here. All right, so like Connie mentioned, I won't have time to present everything. Hopefully everything is displayed in the right orientation, but I will share with you today some of the work of our collaborators and students on two kinds of gene editing methods that are especially.
So David, I think your display, at least for me, I'm getting the wrong display.
Oh,
Switch
okay.
displays.
Is that better, or is that letterboxed? No, I ever since I've used this second monitor, I've had this issue. Let me.
Yeah, we see all your slides, you know, on the left side.
Yeah, we're not seeing the full.
Yeah, the full presenter view.
Okay, I'm gonna have to experiment after this talk with how to fix this without having to stop my talk and unplug my monitor, which is currently my solution. Okay, hopefully this is better. So I'll talk to you today about two methods, base editing and prime editing, that are particularly useful editing methods for correcting genes. Oh, and somebody just pointed out my video is off too. Okay, you have to forgive me. I've only been doing this for 2 years now. Okay, so this slide shows a distribution of known pathogenic human gene variants, and as you can see, they come in all sorts of different flavors. Point mutations are about half of them on the right. There are insertions and deletions and duplications, etc. And so a longstanding goal of life sciences has been to develop ways of installing or correcting as many of these different kinds of pathogenic mutations so that we can study or potentially treat the resulting diseases. Connie covered a really beautiful set of examples where loss of function can often be rescued by introducing healthy copies of a gene. But in many cases, we also need to fix the pathogenic mutations. And all of you, I'm sure, are aware of the beginnings of the modern era of gene editing, which really began with programmable nucleases, such as zinc finger nucleases, TALENs, and of course, CRISPR-Cas9. And these approaches first initiated the current excitement around gene editing by enabling targeted gene disruption. But for reasons I'll explain briefly, it is difficult to apply programmable nucleases to the potential treatment of the vast majority of pathogenic human gene variants. And the reason is that most of the time when you cut the DNA double helix, the resulting end joining processes that the cell uses in response to that double-stranded break result in mixtures of insertions and deletions that we can't control. These are collectively called indels. And they can efficiently disrupt genes because most of the time the resulting indels will cause frame shifts in the gene. So if you disrupt a coding sequence this way or destroy a regulatory sequence that's required for the production of a protein, you can end up disrupting the function of that gene. But in many cases, the double-stranded break is also accompanied by outcomes that are undesired. We now know that chromosomal translocations, for example, and large deletions, aneuploidy, p53 activation, chromothripsis, are all natural consequences of making double-stranded breaks in mammalian cells. And has been recently pointed out in a nice paper by Tony Catherman last year, even if you have a perfectly specific nuclease that cuts only the on-target sequence, simply because mammalian cells are diploid, that nuclease most of the time will actually make cuts in at least 2 chromosomes, and you can measure the frequency of acentric and dicentric chromosomal translocations that result even from a single on-target cut, simply because there's 2 two chromosomes that contain the target sequence. Now, in the presence of a donor DNA template, some really seminal work by Maria Jason and other scientists showed that a double-stranded break can also stimulate homology-directed repair, or HDR. And HDR, because it replaces DNA sequence around the double-stranded break with a template of your choosing, has the potential to precisely correct, not just disrupt genes. But unfortunately, we now know that the efficiency of HDR in most therapeutically relevant cell types is very low. And of course, HDR products are still accompanied by the normal consequences of double-stranded breaks. And as a result, if you look at the clinical trials, the therapeutic clinical trials that are underway currently, there are a lot of them that use CRISPR, virtually none of them are trying to correct a gene. They're almost all disrupting or knocking out a gene. But the challenge, of course, for treating most genetic diseases is that they really the patients stand to benefit a lot more from gene correction than from gene disruption. There are exceptions, and you've probably read about some of the amazing advances in using targeted gene disruption to treat diseases like sickle cell disease and transthyretin amyloidosis. But for most genetic diseases, target gene correction is really needed to maximally benefit patients. And so that inspired our lab to develop base editors a number of years ago now. These are engineered proteins that directly perform chemistry on individual base pairs at targeted sites in the genomes of living cells. Base editors cannot make all possible kinds of edits. They can make 4 kinds of changes efficiently and reliably, which is C to T, G to A, A to G, and T to C. Some of them use CRISPR-Cas9, a disabled form that no longer makes double-stranded breaks, but can still bind to target DNA sites in a guide RNA program manner. Other forms of base editors are CRISPR-free, and they use, for example, tail proteins or even zinc fingers to engage a target DNA sequence. And those CRISPR-free base editors are especially important because researchers have not yet been able to get guide RNAs into mitochondria. And therefore, these CRISPR-free base editors that we reported recently enabled researchers to make, for the very first time, purposeful changes in the sequence of mitochondrial DNA in living cells. And researchers have used these CRISPR-free base editors in animals now as well. All right, so I wanted to summarize an example of using a base editor ex vivo to treat an animal model of a human genetic disease, and then I'll cover an example in vivo. So the ex vivo example I'll tell you about is one that Connie already touched on, which is sickle cell disease. Sickle cell disease is the most common deadly genetic disorder. It's caused by an A to T point mutation in the adult hemoglobin gene, HBB. And there are a number of ways that researchers have taken to tackle sickle cell disease. One of those approaches Connie's already described, which is using a lentivirus to overexpress a healthy non-sickling globin gene. But the approaches that are underway now, which include using nucleases, even trying HDR ex vivo, using lentiviruses, they all have different strengths and drawbacks. And what we were particularly interested in collaboration with Mitch Weiss's lab at St. Jude, was developing a potential therapeutic strategy for treating sickle cell disease that actually corrects the mutated sickle allele, so it stops making the harmful hemoglobin, does not require double-stranded breaks, and does not require an endogenous DNA template, which can create challenges in terms of delivery and manufacturing. So we took an adenine base editor and developed one that was bespoke to correct the HBBS, the sickle cell mutation, not back to wild type. That would require an A to T mutation, which a base editor cannot do yet. But we can change this valine codon that causes sickle to an alanine codon, which is a naturally occurring benign form of hemoglobin called hemoglobin Makasar. And to develop a base editor to efficiently make this correction, we evolved both the Cas9 half of the base editor and the deaminase half of the base editor. To make a long story short, I'll just say that the resulting bespoke base editor can park itself precisely at this CACC PAM, which is a very different PAM sequence than the starting Cas9 recognizes, which is NGG. And using this bespoke base editor, delivered by electroporation ex vivo into the hematopoietic stem and progenitor cells that Connie mentioned, these are from sickle cell human patients. We were really happy to see 80% average conversion of the pathogenic mutation back to this, or I should say to this benign hemoglobin Makasar variant with very few indel byproducts or other unwanted outcomes because we're not making explicit double-stranded breaks in cells. When we transplant these edited human hematopoietic stem cells back into mice, we can then wait 16 weeks and then harvest the bone marrow from these mice. After 16 weeks post-transplantation into an irradiated mouse, it's thought that all of the remaining viable blood cells have pretty much all come from the engrafted hematopoietic stem cells that were transplanted into the irradiated mice. And first we looked at all the lineages that come out of these hematopoietic stem cells and asked whether editing skewed the lineages as evidenced by enrichment or de-enrichment of editing percentage, and we didn't see any. So that suggests that the base edit seemed to be fairly innocuous in terms of differentiation of these hematopoietic stem cells to their downstream lineages. We also use single cell analysis to determine what fraction of the treated human sickle cell patient hematopoietic stem cells were actually edited. And we saw that 87% were edited in at least one, if not both alleles. And this leads to a dramatic conversion of beta globin from essentially all sickle globin to now mostly the benign Makasar globin. And we also see a dramatic decrease of about 5-fold in the propensity of the resulting red blood cells to sickle under hypoxic conditions. We repeated basically everything I just showed you, but this time transplanting mouse hematopoietic stem cells into mice. And the reason is that human red blood cells don't last very long in circulation in mice, but of course mouse red blood cells are fine. So if you repeat these experiments with a humanized mouse model of sickle cell disease, transplanting the hematopoietic stem cells from these mouse models into recipient mice that have been irradiated, you can now compare after 16 weeks how all of the blood cell parameters and other disease phenotypes of these mice compare when they receive edited cells versus unedited cells, or non-transplanted mice that are heterozygous for sickle cell, which these mice are more or less healthy, just like their human sickle cell trait heterozygous counterparts. And what we observed is, even though we used protein delivery in this case, so we got slightly less efficient editing was still pretty efficient, more than 50%. The amount of beta globin Makasar produced was very healthy in the edited mice. And we observed restoration of all the blood parameters that we assayed back to normal levels, consistent with our healthy control heterozygous mice. And we also looked at sickling, the fraction of red blood cells that undergo sickling under hypoxic conditions drops dramatically in the mice receiving the edited cells. The vast majority of the blood cells that come out of the immature blood cells that come out of these mice have the edit. And we also rescue splenic pathology. So one of the hallmarks of sickle cell disease in mice and in young human patients is enlarged spleen and red blood cell pooling in the spleen, which comes from extramedullary erythropoiesis. And we can rescue both of those phenotypes to a large extent in the mice that were given these edited cells. So in summary, ex vivo editing of either human or mouse hematopoietic stem cells that contain the sickle allele, followed by transplantation into animals, seems to rescue all of the parameters that we assayed of sickle cell disease, either completely from the blood parameters or partially from these splenic parameters. Now, ex vivo editing of hematopoietic stem cells takes advantage of a fairly unique property of the bone marrow, which is that it can be taken out of the body, edited, and then put back into the body. That's not something we can really do for the heart or the brain, for example. So the field is also very interested in developing ways to perform gene editing in an animal, in vivo. And an example that we recently completed in collaboration with NIH and the Progeria Research Foundation is the use of base editing to treat this rapid aging disease, progeria, which is caused by a single C to T mutation in just one copy of a gene called human lamin A, which results in the production of this toxic progerin protein, which damages nuclear membranes and causes premature aging and death on average at about 14 years old in people. This is actually maybe a good opportunity to also point out that this is an example, progeria is an example of a disease that can't be effectively treated by gene complementation therapy, meaning putting in a virus, for example, that makes more of the normal lamin A protein, because the problem is not a lack of normal lamin A. The problem is that this progerin protein is toxic. So we really need to get rid of the toxic progerin protein. And you might say, well, maybe you could disrupt it using a nuclease, and researchers have tried that, but that approach faces a number of challenges, including the fact that you need to distinguish between the pathogenic allele, which you want to disrupt, and the healthy allele, which is only one base pair different, which you absolutely don't want to disrupt. And there's also the possibility, because as I showed you, when you cut with a nuclease, you make a whole mixture of dozens or hundreds of gene products, that some of those products you make could themselves be pathogenic. So we developed instead a base editor that directly converts this T back into the wild type C. And we first did studies in cell culture where we made a lentivirus that infects patient progeria fibroblasts with this adenine base editor that was tailor-made to make this correction. And we were happy to see about 90% correction of the pathogenic allele back to the wild type allele in either of these two patient-derived fibroblasts. And those and other studies I don't have time to present were encouraging enough that we initiated a series of long-term animal studies in collaboration with NIH and Vanderbilt. As Connie mentioned, adeno-associated viruses have cargo limitations, and it turns out that at least our previously published versions of base editors are too large to fit into a single AAV. We now have unpublished single AAV base editors that work very well. So we hope to get those into the hands of the community soon. But at the time we did the study, we had to divide the base editor into two halves, which we, to each half, we fused a little piece of protein Velcro, a trans-splicing intine. And then we injected sort of therapeutically useful levels of AAV9, or I should say therapeutically tolerated levels based on current human AAV drugs and human clinical trials, injected these AAV as a pair into progeria mice that Francis Collins's lab had generated, which have two copies of the human mutated lamin A gene. These mice age rapidly, have cardiovascular problems, just like the progeria children do, and die young. And in these mice, we were happy to see that, especially in the sort of better edited mice, the ones that were large enough at the time of injection that we could give them a full dose, we got pretty good editing in a variety of tissues. For example, 25 or 30% editing in the heart, 60% editing in the liver, et cetera. And the consequence of that DNA editing, which was accompanied by relatively few indels, again, because we're not cutting chromosomes, was rescue of progerin at the RNA level. So we see a sharp decrease in the amount of the misspliced progerin protein, the misspliced progerin RNA, as well as a sharp decrease in the amount of the undesired toxic progerin protein. So here's a Western blot where every lane represents a different mouse. And you can see that even though we're only getting in the heart maybe 30% correction at the DNA level, we're seeing more than 30% rescue at the protein level. We don't rigorously know why that is. I can talk about it maybe in the discussion, but it was certainly welcome to see. And then I'm skipping over a lot of other studies we did and just jumping to longevity. So we had two cohorts of mice. The younger mice, the mice that were injected when they were younger, received 10 times less virus, but nevertheless, their lifespan was extended 1.8 times that of saline injected counterparts. And the better edited mice that received the full dose, they reached a median lifespan of 510 days, which is about the start of old age, even in normal non-progeria wild type mice, and is about 2.4 times the lifespan of the saline injected control mice. So this level of lifespan extension in an animal model of progeria has never before been observed. And then here are movies of what progeria mice look like. First, an untreated mouse, about 7.5 months old. So this is about as old as these mice get before they die. And you'll see that the mouse has a very low activity level. It has extensive outward curvature of the spine or kyphosis, a very thinned and whitened coat. And this mouse is, as you can see, pretty close to the end. But these are progeria mice, same genotype. These are 11-month-old females. So they're far older than any untreated progeria mice can live. And they've received a single injection when they were 14 days old of our base editor adeno-associated virus. And you can see they look pretty much normal. They're a little bit smaller than wild type mice, but otherwise fairly normal. And likewise, here are 3 P14 injected AB treated male progeria mice, plus 1 wild type C57 black mouse, which is the slightly larger mouse here. And also at 11 months old, and you can see these mice are also doing much, much better than they would even at 7 months if they were untreated. So we're currently working with the NIH and the Progeria Research Foundation and Beam Therapeutics to try to bring this treatment towards progeria patients. All right, so that's an example of ex vivo and in vivo therapeutic base editing in animals. I wanted to highlight some other work that's moved base editing closer and closer to human patients. The scientists at Penn and Verve Therapeutics and Beam Therapeutics reported last year some really exciting results using an LNP, which Connie also introduced, to deliver an adenine base editor messenger RNA into the liver of monkeys, targeting PCSK9, showing that they could durably and very potently lower LDL serum cholesterol as a result of achieving about 60% base editing in these animals, in the liver of these animals. Just in November, last November, so about 5 months ago, Beam Therapeutics received FDA clearance to proceed with the first human clinical trial of a base editor. That occurred about 5.5 years after we published the very first paper describing a base editor, which is much, much faster than I thought it would, and is really a testament to the fact that literally thousands of laboratories are using base editors. Our base editor constructs have been sent out by Adgene more than 11,000 times to more than 3,000 laboratories around the world, and there are now more than 600 base editor papers that have been published, base editing papers. So I'll end with a little bit on prime editing. So base editing corrects the sort of blue wedge of this pie chart, the transition point mutations, but that leaves a lot of mutations that base editors can't directly correct. Now, some of them you can still address, at least at the disease level, by using the base editor to disrupt a gene or to make a benign mutation like we did with sickle cell, or to change a regulatory sequence. But of course, we're particularly interested in developing ways of directly correcting back to the normal sequence insertions, deletions, transversions. And so in late 2019, we reported prime editing, which is a different kind of mammalian cell gene editing method, also programmable, that allows you to make just about any kind of local change to a sequence of target DNA in a living cell. And the way it works is the prime editor is a fusion of a Cas9 nickase with an engineered reverse transcriptase. It nicks the target DNA sequence just upstream of the desired edit. And then a specially engineered guide RNA called a PEG RNA that not only encodes where to target, but also what the edit should be. The edit itself is encoded in a 3' extension in the PEG RNA. That PEG RNA works with the freshly nicked DNA and the engineered reverse transcriptase to initiate reverse transcription of the template that is in the guide RNA, in the PEG RNA, extending this nicked 3' hydroxyl of the nicked target DNA strand. So you'll copy base by base the sequence that's in the PEG RNA extension. And then thanks to the dynamics of DNA repair in mammalian cells, actually in many cell types, you get this flap exchange followed by flap cleavage and ligation. And in our sort of PE3 prime editing systems, we nick the non-edited strand, which biases replacement of that strand using the edited strand as a template. And that's how you can get a permanent prime edit in both DNA strands. And you can completely control the sequence of this reverse transcriptase template. So you can make it a little bit shorter or a little bit longer than the sequence it replaces. And that's how you can make deletions or insertions in addition to substitutions. And you can now make all 12 possible base-to-base changes at targeted sites in the human genome. These are in human cells. You can make precise deletions or insertions or mixtures thereof. And so our lab and others have used prime editing to now install or correct a wide variety of human pathogenic alleles. You can now install the sickle cell mutation, making a homozygous triploid, in this case, sickle cell cell line, and then you can take those homozygous sickle cells and use prime editing to correct them back to wild type, again, with high ratios of correction to indels. Likewise, you can, for the first time, make a precise 4-base insertion at the site in the human genome that causes Tay-Sachs disease. And then you can use prime editing and cleanly remove those 4 extra bases, returning it back to wild type. You can install disease prevention alleles. So this is a pathogenic allele all of us have, which is our prion protein gene. And if you make a G to T transversion at the right location, you can protect from prion disease. This mutation actually evolved naturally in a colony of ritualistic human cannibals. And we showed that you could install this with prime editing with a good ratio of editing to indels. And you can insert a variety of sequence tags, including recombinase, site-specific recombinase landing sites, which then allow you to subsequently, after prime editing to install the site, you can use those recombinase sites and install whole genes or make very large inversions, something we published recently. So we focused a lot of recent effort on improving prime editing systems and understanding how they work in greater detail. And to make a long story short, I'll just summarize how we've improved PEG RNAs. First, by recognizing that the extension, the 3' extension on the PEG RNAs that contain the critical edit in red here, that's a real vulnerability for prime editing systems because that extension is prone to degradation. And if you degrade that extension, you don't just prevent that PEG RNA from mediating prime editing. You actually poison prime editing in the entire cell because that PEG RNA degraded can still occupy the prime editor protein and can still occupy the target site. And so we showed this explicitly by spiking in synthetically truncated PEG RNAs and showing that they have this very deleterious effect on prime editing. So we discovered structured RNA motifs that, which we call result in engineered PEG RNAs or ePEG RNAs, which improve the stability of PEG RNAs. And we showed explicitly by capturing using terminal transferase a snapshot of all the prime editing intermediates in a human cell. We showed that using these engineered PEG RNAs greatly increases the fraction of PEG RNAs in a human cell that at any instant are still fertile for doing prime editing. And so just making this simple change of adding a secondary structure to the 3' end of PEG RNAs can greatly increase the efficiency of prime editing in a wide variety of different cell types, including some primary patient-derived fibroblasts. And so using these ePEG RNAs is what we now recommend and what we do for all of our prime editing experiments. Now, on the prime editing protein side, we did a collaboration with Jonathan Weissman's lab and Britt Adamson's lab to first identify what are the cellular determinants of prime editing outcomes. Britt's lab has set up a really wonderful CRISPR-I screening platform called RepairSeq, in which they've knocked down each of almost 500 DNA repair genes. And by carefully designing the experiment so that the prime edited site is located within one sequencing read length of the guide RNA that programs that CRISPR-I screen, we can simultaneously sequence each CRISPR-I guide RNA to identify what gene has been perturbed in that cell and identify all of the ways that the cell's prime editing outcomes have been changed as a result. And to make a long story short, we consistently got the answer that the strongest hits to come out of the screen were genes that were implicated in mismatch repair. And if you knock down those mismatch repair genes, you increase the efficiency of prime editing and you decrease the frequency of indel byproducts that come from prime edits. This actually makes sense because mismatch repair in humans is nick directed, and the intermediate in prime editing that you make first has the edit on just one strand. And because that strand had to be nicked in order to promote reverse transcription, the edited strand with the nick is a substrate for mismatch repair that if mismatch repair gets to this intermediate, will revert that sequence back to the starting sequence or produce indels through the process of repairing this nicked piece of DNA. So we wondered whether we could transiently inhibit mismatch repair long enough to give this nick a chance to be ligated, which would then prevent mismatch repair from selectively reversing our prime edit. So we developed an engineered dominant negative form of MLH1, which we call MLH1-DN, and showed that if you transiently express that MLH1-DN dominant negative protein, you can increase quite a bit, in this case, threefold or more on average, the efficiency of prime editing. And so that led to our current generation prime editor systems, PE4 and PE5, which are basically PE2, the simple form of the prime editing system that does not nick the unedited strand. That plus MLH1-DN is PE4, or you can use the full-blown version of the prime editing system, which nicks the non-edited strand. That's called PE3, as I mentioned. If you co-express transiently MLH1-DN, you get PE5. And we showed that on average, PE4 and PE5 increase the efficiency of prime editing quite a bit, especially in cell types that are competent for mismatch repair, which is most cell types. Interestingly, hex cells are not. They're somewhat mismatch repair deficient. And it's tempting to speculate that if we had tried to develop prime editing in, for example, HeLa cells instead of hex cells, we may not have gotten very far, or we would have been much more discouraged. It just happened that we were fortunate to have chosen a cell line to develop prime editors that were naturally mismatch deficient to begin with. So we saw right off the bat quite efficient prime editing. These improvements also apply to more therapeutically relevant cells like these human iPSCs. And then maybe my favorite part of this recent work is a free lunch you get from just recognizing how mismatch repair influences prime editing. So it's a nice example of how basic science insight can lead to a real benefit. And that is, we understand from the literature that mismatch repair does not engage sites that have a lot of mismatches. So mismatch repair systems engage single mismatches, double mismatches, maybe some triple mismatches. But once you start putting in a lot of mutations at the mismatch site, the mismatch repair machinery doesn't see it as a mismatch anymore. Other DNA repair machinery has to deal with it. So we wondered whether we could just install benign silent mutations near the desired coding mutation we wish to make, shown in green, and increase the efficiency of prime editing. And that turns out to be the case. You can sometimes double or even more the efficiency of your prime edit simply by counterintuitively adding additional mutations beyond the mutation that you wish to install. And that's really an insight that came about from this deeper understanding of how the cell is responding to prime editing. And if you combine all of these advantages, optimizing the architecture of the prime editor, which I didn't get time to talk about, the PE4/5 system and the EPEG RNA system. These approaches, these benefits all synergize because they all address different bottlenecks in the prime editing process. And collectively, we can now see sometimes 10 or 100-fold improvements in prime editing by combining all of these improvements shown in this graph. And so we now recommend that people use the PE4, PE5 max system with the EPEG RNAs to do their prime edits to begin with. All right, so to summarize, base editing can make the 4 kinds of transition mutations pretty robustly now. You can do so using all protein CRISPR-free base editors that enable you to edit mitochondrial or chloroplast DNA. And I showed you that ex vivo, you can use a base editor followed by transplantation into an animal to rescue sickle cell disease in mice. In vivo, you can use a base editor to rescue progeria in a mouse, all using the kinds of delivery methods that Connie outlined in her talk. And finally, I summarized for you the development and recent advancements in prime editing, which uses a nicked target DNA site to directly write edited DNA sequence into the human genome at a specified site. And so that just leaves me to thank a very talented and hardworking group of students and postdocs and collaborators who made all this work possible. Andrew Anzalone led the development of prime editing. Peter Chen, in collaboration with the labs of Britt Adamson and Jonathan Weissman, led the development of the PE4, PE5 system. Nicole Godelli and Alexis Comor developed the adenine base editor and the cytosine base editor, respectively, back in the day, as we say. Luke Koblen, in collaboration with our collaborators at NIH, Francis Collins and Mike Erdos, and at Vanderbilt, Jonathan Brown, and the Progeria Research Foundation. Leslie Gordon led the progeria work. Beverly Mock, in collaboration with Joseph Mogus's lab and Vamsi Mutha's lab, led the development of the CRISPR-free mitochondrial base editors. James Nelson and Peyton Randolph led the development of the EPEG RNAs, and Greg Newby, in collaboration with the labs of Mitch Weiss and Shendar Tsai and Chandra Pruitt-Miller and others, led the development of the base editing treatment for sickle cell. And grateful to these agencies for funding, and I'm happy to start our discussion.
Thank you so much for the wonderful talks. For the questions, I just wanted to thank everyone who submitted a question when registering. We'll read out a few of those, but please feel free to ask your questions live. You can raise your hand, type questions in the chat box. We have a question from Annie to get started.
Oh, hi, I have a question for Dr. Liu. So how's the gene editing, or particularly like the prime editing example, can that be applied to histone tail mutations?
Meaning mutations in, sorry, what do you mean by histone tail mutations?
Like basically the tail on the, like for example, like I'm from the brain tumor group, and I just wanted to know that, say for the H3K27M, would that be a possible application for that that approach?
Yeah, so if you mean editing the genes encoding the steps that cause those methylations, it's possible, but of course, histone methylation is involved in a wide variety of biological pathways. So I think if you want to tinker with histone methylation, you know, it's possible as a basic science study, probably not as a therapeutic approach. I thought you were asking a slightly different question, which is what's the effect of chromatinization on gene editing? And you know, the answer to that question is it does have an effect, and generally sites that are highly chromatinized are harder to edit. But there are gene editing technologies that also seem to have at least some ability to edit chromatinized DNA. And there are a variety of strategies researchers have developed to also try to work around that issue.
Thank you.
Sure.
Thank you. So I'll read one of the questions from registrants. We had a couple of people asking about the challenges of delivery in the brain and the prospects for targeting specific cell types. And I think you spoke about that a little, Connie, but I was wondering if you'd like to comment on that more.
Sure. So I think that is one of the more challenging injection or targeting sites. And there's a couple of questions that I think you have to consider. One is how many neurons and where are they? If you're trying to get a very specific area transduced, it's possible you can directly inject. Now for humans, that's challenging. But in fact, I think for Parkinson's disease, they drill a hole in the skull and stick a needle in. And I think they've done up to 8 sites to try to target for Parkinson's, target one area for Parkinson's. There was some problems with that in the clinical trial. I don't know the details. They said there were MRI abnormalities. If you're doing this for an experiment where you want to get it into a particular place in a mouse, that is an approach you can use, and people inject AAVs into specific areas all the time using stereotactic injections. But if you were going for something that was more broadly disseminated, lots of different areas in the CNS and the brain, that's a really tough one, but it's been approached by engineering the capsid. And there's a very nice study that was out of Viviana Gradino's lab by Ben Deverman, who used a screen to find capsids that could cross the blood-brain barrier. So you could do a systemic injection, a retroorbital injection, and those capsids somehow evolved the ability to get out of the blood vessels and into the neurons. And you can get very good coverage of the brain with that method in mice. That same capsid doesn't work in primates. And so people are now trying to do the same kinds of selections to find capsids that would work the same way in primates, that is, get widely disseminated infection from a systemic injection. I haven't seen any data on that. I understand it's happening. So really you have to ask where and how many cells do you need to transduce for the therapeutic effect? And then, you know, what approach you need to use. I think it's a tough one. I think you have to deliver a lot of virus, which can then incite some cytokine release, which could be deleterious. So when you have a large number of cells and they're disseminated, that's where you start to sort of tread a line with toxicity. But maybe more efficient capsids will allow you to get over that hurdle and get some therapeutic effect. A secrete, I should say one more thing, a secreted product, again, might be okay. If you're looking for something like a growth factor secretion, where again, you don't have to have a high percentage transduced, and that could be a good strategy for certain types of degenerative diseases. If you know what secreted growth factor or product might be capable of rescuing many more cells than are transduced. Does that answer the question? Do you know? I don't know who asked the question.
I'm not sure who asked it, but I think that was very helpful. Thank you.
Mhm.
So we have a series of questions. One big picture question I thought was interesting was, what stage of gene therapy development for a particular disease might you think about working with a company, you know, versus an academic lab? What are the key considerations, the pros and cons? I guess, like, what's the stage when you think about making that transition?
I mean, David might be able to answer that because he's gone further. I can say what we're doing. We do a lot of proof of concept in mice. And once we have vectors that work in mice, we go to companies and ask if they want a partner to take it forward for safety in monkeys and then on to clinical trials. So we're hoping to do that very soon. It's a lot of money to do those kind of experiments. So I think the basic science is something you do in your lab, the preclinical work. And then once you get something to work, usually in mice, you need a lot more money, and primates is the way to go. And that is when you look to partner. At least that's what we're doing. David, again, may have more experience and different answer.
No, I think it's my answer is actually similar. I think the bottom line is it's very hard, almost impossible, I'd say, for an academic lab to take experimental treatment all the way into the clinic. Just from a resource and expertise in manufacturing, barriers are very high. So like Connie, I think the blueprint is to show proof of concept in cells and then in mice. Occasionally, academic labs can do studies in monkeys. It's very, very difficult, both from an expense point of view, you know, it's very slow as well. And there's ethical questions, for example, and there's animal availability questions. For example, in progeria, monkey models were never made until recently. And now that there's a couple monkeys that have been made and characterized, if we want to generate enough to do a study, well, how long does it take to make a monkey? Well, to a first approximation about as long as it takes to make a person. So it's not super easy to just breed up a colony of monkeys. And of course, the expense and the ethics of doing so is also can be challenging. So like Connie, we take it as far as we can in animal models, and then try to present enough proof of concept data that's compelling that companies are interested in taking it to people. And I think, to the credit of many companies, often it's not a question of whether they expect to make money off of the program. I mean, obviously, ultimately, companies have to make money to stay afloat, but especially for these early experimental therapies, I think a lot of companies really do see special importance and responsibility and purpose and value, therefore, in helping to pave a way forward for new technologies that could become broadly applicable to treat many diseases.
Thank you. So we have a lot more questions from that people had submitted, but does anyone want to ask a question live before I? Okay, if not, then let's see. One of the questions is about interpreting results in humans. So if patients survive, as one would hope, after experimental gene therapy, it of course wouldn't be possible to do postmortem analysis, and the clinical readouts might be very indirect. That will presumably make it difficult to interpret the results of clinical trials for CNS gene therapies. How might this be overcome? What biomarkers might you use to measure target engagement?
I mean, a lot of that is going to depend on the disease. I think the disease particulars. So, before you do a clinical trial, you have endpoints that are the goal of the study. And certainly those would be evaluated during the trial. Post-mortem then, I mean, one of the things we're hoping to do is to track where did the virus go using some in situ methods. So we can detect a single viral genome using fluorescent in situ methods. So at least we're hoping to recover tissue where we can look at where did the viral genome go. And then you can also assay, is there viral gene product made using immunohistochemistry or RNA fish? So you can at least track, I hope. I mean, it's not easy to do that systematically. The human brain is huge, for example, if you're targeting the brain. But you may have certain areas you're most interested in, and you can at least see what's there. And that might enable you to say, okay, the virus did end up here. The products were made. And then relating it to biomarkers or whatever is going to depend entirely on the disease that's being addressed. There has to be some real thought about that before you go forward, I would say. But yeah, I think it's all disease dependent.
Thank you. So we have a couple more. One is interest in the panel's view on clinical trial results from the EDITAS trial. Yeah, they're just asking for some comments, saying that they read the initial efficacy results were less dramatic than hope. What lessons can be learned? Do you have any comments to share?
I only know what's been reported publicly, which was that they had a small number of patients, and it was an AAV delivered Cas9 nuclease or Cas12 nuclease to disrupt a sequence that results in exon skipping that I think restores the function of the gene. And I think they observed some efficacy in some of the cohorts, but both because there was a small number of patients and efficacy was not observed across all groups, the results were considered sort of a moderate success as opposed to a home run. That's about all I remember, but it's also Connie's field. She has more field expertise than I do, so she may have other comments.
Yeah, I haven't looked exactly at the patient population, but I would say that a lot of these degenerative diseases have an issue of sort of a point of no return. So you need to do the therapy while you still have healthy enough cells to save. And I know that a lot of diseases, for safety reasons, people are not treating a well-sighted eye. They tend to, in the initial trials, to go into an eye that doesn't have that much vision, so they're not risking loss of that vision. And so if that's a stage where there aren't enough cells left to save, or the disease process has advanced to the point where you can't save, then that's a reason why you might not get a home run. And it was a small number of patients. It is a rare disease, and I don't know what age they were treating. But if you look at the other ocular trial, the Luxturna, they've been moving to earlier and earlier, younger patients, including, I think, 2-year-olds now. And it's for the same idea that you want to be able to do the therapy while the architecture of the eye is still good. The cells that you want to rescue are still able to be rescued. And that's the sort of thing that might happen with LCA10. Again, I don't know if there's enough patients that are even that are available, that are in the different age groups, but I think that's tends to be a problem with some of these gene therapy trials as you go to late stage diseases, because the risk is lower, but you then don't get efficacy.
Thank you. And I think for one big picture final question, I wanted to ask about what you're both most excited about, as you're thinking about the next 2, 3, 4 years, what has the most potential? What are you really excited about?
David, you must be excited about a lot of your gene editing going on. That's a good one.
Yeah, one decision I made in putting this talk together was whether to talk about some of our engineered VLP work, virus-like particle work that we just published. I'm excited about. Connie introduced the topic beautifully. There's basically retroviruses in which you've replaced the viral genome with your desired cargo. And for gene editing agents, proteins are really the ideal cargo, because if you're editing the genome, you just want to get in quickly, edit the on targets, and then get out. And the off targets tend to be edited slower than the on targets. So the longer you stay, the worse the specificity. Plus, you never want your intervention in the patient longer than it needs to be. And really the whole promise of gene editing is that you can do a one and done acute treatment that results in a lifelong benefit. So using engineered virus-like particles, we've delivered base editors and nucleases into animals, and we can actually see really, in some cases, very efficient state-of-the-art levels of editing, but without ever having to introduce DNA or messenger RNA. And we do observe virtually no off-target editing when we deliver proteins with these eVLPs. So I'm excited by that, and by another concept that Connie introduced in her talk, which is that because these are retroviruses, you can swap out the glycoproteins on the envelope to different glycoproteins to change the cell type tropism of the resulting virus-like particles. And so that's an area that we're really excited about and have rapidly been investigating.
And I'll also address this a little bit from the perspective of our own work. As Perry mentioned in the beginning, we've been looking for therapies where we can rescue vision across a large number of genes that affect vision. So I think the number of genes that can cause vision loss is in excess of 250. So addressing each one individually with any type of specific gene editing, gene augmentation, et cetera, is really tough. Many of the patient families are small, and it costs a lot to develop these therapies. And logistically, it's just tough to imagine doing 250 tailored types of approaches. So rather than that, we're trying to find generic problems that cut across these different genotypes that we can address with generic solutions. And so it's pretty clear in almost every degenerative disease that I know of that there's oxidative damage, there's inflammation, there's metabolic derangement. So we've been addressing each of those using the eye as a model system. And we now have, I think it's 6 genes that will address these different problems. And so we're trying to see if we put them in combination, will we be able to prolong vision? And we can do so individually in mice across different disease genotypes. So we know in mice, it's somewhat of a generic, at least, solution. And we're thinking combinatorially, we might get a bigger boost because we'll be addressing the multiple problems that we think exist. And so for us, we're really excited that some solutions from gene therapy might at least extend vision, if not truly correct in a long-term fashion, at least extend vision for people who are losing their vision. And I think we all know how precious vision is, and just if we can extend it for a couple of years, maybe even more, hopefully, that might be a really big win, even if we can't cure the disease.
Thank you. Thank you both. It's really incredible to hear about some of these future directions and hopes. So I think we're out of time. So thank you both again for your wonderful talks, and thank you to everyone who attended.
Thank you.
Thanks, everybody.